In this video, we learn more about how genes work. The work of early geneticists illuminated the structure and function of DNA and genes, as well as the method in which these genes were inherited from one generation to the next. But biologists still did not understand how gene expression occurred. How was the information contained in the genes expressed? Gene expression is the process of translating the information in DNA into functioning molecules within the cell. It was already understood that genes carry the instructions for making and maintaining an individual. George Beadle and Edward Tatum proposed that if you damaged a gene by creating a mutant and observed the resulting effect on the mutant's phenotype, meaning the physical traits that the organism develops, this would indicate what the gene does in an organism. So it would indicate the gene's typical function in an organism. The mutants that they created are now known as knockout, null, or loss of function alleles. To test their hypothesis, Beadle and Tatum damaged genes in the bread mold Neurospora crassa. The products of these genes were involved in particular reactions in a metabolic pathway inside Neurospora crassa cells. They observed that defects in particular genes resulted in the mold's inability to produce specific proteins. On the basis of these results, they concluded that one gene is always responsible for making one enzyme. Since the genes that they were studying were enzymes that were part of a metabolic pathway, they thought one gene might be responsible for the production of one enzyme. This hypothesis has now been revised to be known as the one gene, one polypeptide hypothesis, since genes not only code for enzymes, but other protein molecules that serve several different functions inside the cell. For example, proteins that are part of the cytoskeleton, proteins that serve as transport channels within cell membranes, so on and so forth. What came to be known as the central dogma of molecular biology was first proposed by Francis Crick. He proposed the idea that since DNA is an information storage molecule, the sequence of bases in DNA serve as a kind of code, and different combinations of these bases would specify 20 amino acids that make up proteins. He also went on to propose that a particular stretch of DNA, a gene, contains the information to specify the amino acid sequence of one protein. The information encoded in the base sequence of DNA is not directly transferred or translated into an amino acid sequence of proteins, but something happens between DNA to when it becomes a protein that needed to be figured out. It was Francois Jacob and Jacques Monod who proposed what happens to the information between DNA and proteins. They proposed that RNA molecules were the ones that acted as a link between genes found in the cell's nucleus and the protein manufacturing centers located in the cytoplasm. A specific kind of RNA called messenger RNA was found to carry information from DNA to the site of protein synthesis. The enzyme RNA polymerase makes RNA or synthesizes RNA according to the information that it derives from the sequence of bases in a particular stretch of DNA. In eukaryotic cells, DNA is present within the nucleus of the cell. This information gets first transferred to an mRNA molecule within the nucleus by the enzyme RNA polymerase. Then this mRNA travels outside the nucleus into the cytoplasm of the cell, where it takes the information to the site of protein synthesis, which is the ribosome, and the ribosome translates this information to build protein molecules that perform various functions inside the cell, including catalysis, maintaining cell shape, carrying molecules, and vesicles from one part of the cell to the other, and many other functions. So to summarize the central dogma of information flow within cells, we can say that DNA codes for RNA, which codes for proteins. So the information thus flows from DNA to proteins. And these proteins are the functioning molecules that serve different functions in the cell. The sequence of bases in a stretch of DNA specifies the sequence of bases in an RNA molecule, which specifies the sequence of amino acids within a protein. So genes ultimately code for proteins. The process of information transfer from DNA to RNA is called transcription, and the information is specifically transferred to messenger RNA molecules or mRNA molecules by the enzyme RNA polymerase. The information within mRNA is then translated to form proteins. We know now that information is stored within the DNA in the nucleus of cells. This information is then passed on to an mRNA molecule. Specific sequences within DNA, called genes, store codes for proteins, but this information is first passed on to mRNA molecules, which serve as information carriers that then leave the nucleus and deliver this information to the site of protein synthesis, where this information is used to build protein molecules that serve as the active cell machinery performing different functions inside the cell. So there are two terms that are important to know here. The genotype of an organism is the sequence of bases in its DNA. An organism's phenotype is a product of the proteins it produces. Different forms of the same gene are called alleles, and these alleles differ in their DNA sequence. Proteins produced by these different alleles differ in their amino acid sequence. Let's take a look at what this means. Supposing we're looking at a gene that specifies coat color in mice. Mice have different coat colors. Some forest mice have dark coats. Some mice, like the beach mouse, have light coats. So what brings about this difference? If we look at the DNA sequence of each of these different alleles of the same gene, that makes a protein called the melanocortin receptor, we see that in forest mice, the sequence is slightly different from the one found in beach mice. When these sequences are transcribed to form mRNA molecules, and then when this information is used to build the melanocortin receptor, there are amino acid changes in the protein that is produced. In forest mice, this particular amino acid sequence, this is just part of the gene that we're looking at. There are additional amino acids in this protein that are not shown, but this particular amino acid sequence leads to the formation of dark coats in forest mice. On the other hand, a change in an amino acid for the same protein in beach mice leads to the formation of lighter coats in these mice. The information stored within the DNA is known as the genotype. When the information is transferred to proteins that bring about the difference in these traits, it is called the phenotype of the organism. Although most of the times genes ultimately code for protein molecules, there are certain exceptions. Many genes, for example, code for RNA molecules that do not function as messenger RNAs. There are other kinds of RNAs that are produced within the cell that do not get translated into proteins. Even though these RNAs don't get translated into proteins, they serve important functions in the cell. Another exception is that sometimes information flows in the opposite direction, from RNA molecules back to DNA. For example, some viral genes are composed of RNA, and they use an enzyme called reverse transcriptase, which is a viral polymerase, to synthesize a DNA version of the virus's RNA genes. In this case, information is now flowing back from RNA to DNA. So how does the sequence of bases in a strand of mRNA code for the sequence of amino acids in a protein? The genetic code contains the rules that specify the relationship between a sequence of nucleotide bases in DNA or RNA and the corresponding sequence of amino acids in a protein. The way that scientists worked out how the information in DNA coded for information in proteins, they reasoned that there were only 4 bases within RNA that must specify in some way for 20 amino acids that are used to build proteins. If each base coded for just one amino acid, they would only have a code for 4 amino acids. So that wouldn't be enough because we need codes for 20 amino acids. If a 2-base code was used, then there would be codes enough for 16 amino acids, which is still less than 20. On the other hand, if a 3-base code was used, there would be more than enough codes, 64, for 20 amino acids. So they hypothesized that a 3-base code was used to specify amino acids in order to build proteins. This 3-base code is known as the triplet code. This triplet code is redundant, meaning that some amino acids are specified by more than one triplet code. And this 3-base sequence is called a codon. Each codon specifies for one amino acid. Based on the hypothesis that a 3-base code must be needed for specifying 20 amino acids, Marshall Nirenberg and Philip Leder devised a system for synthesizing specific codons. Using this system, they were able to decipher the genetic code by determining which of the 64 codons coded for each of the 20 amino acids. During this process, they found that there was one start codon that was always used to initiate synthesis of proteins, and this was called the start codon, which contains the 3-base code AUG. This signifies the start of the protein encoding sequence in mRNA. They also found that there are 3 stop codons, UGA, UAA, and UAG, in the genetic code that signal the end of the protein coding sequence. So when the ribosome encounters these stop codons on an mRNA molecule, this signals the end of protein translation. So by synthesizing codons and finding out what amino acids they specified, Nirenberg and Leder found that there were examples where an amino acid was specified by more than one codon. For example, serine here is specified by 4 different codons. There was a start codon that was always present at the beginning of mRNA molecules, and this codon specified the amino acid methionine. And there were 3 stop codons that always signaled the end of protein synthesis. This table is called a codon table that is frequently used to figure out the amino acid sequence given an mRNA sequence. Some important properties of the code include that it is redundant. All amino acids except 2, methionine and tryptophan, as indicated in this image, are encoded by more than one codon. It is unambiguous. One codon never codes for more than one amino acid. It is nearly universal in that all codons specify the same amino acids in all organisms. Again, there are some exceptions in this case. And lastly, it is conservative, meaning that the first 2 bases are usually identical when multiple codons specify the same amino acid. Let's learn how to use this code. If given a DNA sequence, you should be able to transcribe it in the form of mRNA and then translate it in the form of a protein. So if the DNA has this particular sequence, we know that DNA is double-stranded. Both the strands are anti-parallel to each other. RNA polymerase synthesizes RNA in the 5' to 3' direction. Knowing that, we can assume that it will use the bottom strand as a template strand and would synthesize complementary bases to that template strand. Keep in mind that mRNA contains U instead of T, so U would be incorporated in the mRNA sequence. In this case, we're looking at the beginning of a gene, and so our first codon is always going to be AUG, which specifies for methionine. Methionine is always the first amino acid in all proteins. Using the codon table, we should then be able to translate the information within mRNA to build an amino acid sequence. Another thing to note that a stop codon, in this case UAA, always signals the end of protein translation. So take a minute and transcribe and translate the DNA sequence that is shown in panel B of this image. When you're done, you should have the sequence indicated on the image. The sequences of bases within genes can have mutations, which are permanent changes in an organism's DNA. You can think of them as a modification in a cell's information archive that leads to a change in its genotype. Mutations are often able to create new alleles, but there are also mutations that can be harmful. There are different types of mutations. These include point mutations that result from a single base change, and chromosomal level mutations that are larger in scale and often result from the addition or deletion of chromosomes from the individual's karyotype. We are going to focus on point mutations for this session. Point mutations occur when DNA polymerase inserts the wrong base into the newly synthesized strand of DNA. This results in a change in the DNA base sequence. Now, DNA polymerase is usually able and very efficient at proofreading its work and correcting any errors that exist when it synthesizes new DNA strands. But when these proofreading and mismatch repair systems fail, these mutations persist within DNA. Point mutations may be of several types. There are missense mutations or replacement mutations. These result in changes in the amino acid sequence of the encoded protein. Point mutations can also be silent mutations. These mutations do not change the amino acid sequence of the gene product. So when DNA replicates and new DNA is being synthesized by using the existing DNA as a template, sometimes the wrong bases get added on by DNA polymerase. Now, usually DNA polymerase is pretty good at fixing these errors, but sometimes these errors fail to get fixed and result in a completely changed base pair in the new DNA sequence that results in the formation of a mutant gene. Now, mutant genes are not always harmful to an organism's health. They can lead to differences or generate new alleles of genes, but sometimes these mutations can have a negative effect on an organism's health. So if we're given an original DNA sequence which generates an original mRNA transcript that leads to the formation of this polypeptide, if there is a change in DNA from the base T, for example, to the base C, that change is going to be transferred over to mRNA and maybe to the protein. In this case, since the codons UAU and UAC both code for the amino acid tyrosine, this mutation does not result in an amino acid change in the protein. Subsequently, there is no change in the phenotype, and this mutation is neutral with respect to fitness of the individual. These mutations are known as silent mutations. Now, suppose that the base G is changed to the base T, this would lead to a change in the mRNA sequence. If you compare this mRNA sequence to the original mRNA sequence, since the codons UGG and UGU do not code for the same amino acid, this mutation leads to one amino acid change within the protein. This mutation leads to a change in the primary structure of the protein and may be beneficial, neutral, or harmful depending on the role that this amino acid plays in the structure and function of that protein. This mutation is known as a missense mutation. A third kind of mutation, called the nonsense mutation, results in a change in the mRNA sequence such that the codon now becomes a stop codon and leads to an early truncation of the protein. This may lead to the mRNA becoming unstable and breaking down, or a shortened polypeptide as shown in this image. A shorter protein is not going to be able to perform its role in the cell well. Hence, these mutations are most often harmful to an organism's health. And the last kind of mutation that can take place is called a frameshift mutation. This results when an extra base is added to the DNA or an existing base is accidentally removed from DNA. In this case, an addition is shown. The base C is added to the DNA sequence, resulting in its passage into the mRNA sequence. This leads to a change in the frame of the codons, which results in a completely different sequence of amino acids from the original one. Thus, frameshift mutations lead to a change in the reading frame, a shift in the reading frame of the mRNA, altering the meaning of all subsequent codons, and this results in synthesis of a completely different sequence of amino acids past the mutation. Frameshift mutations are most often dangerous to an organism's health. So to summarize the effect that mutations have on organisms, they can be either beneficial and increase the fitness of the organism. In this case, this mutation will persist in a population and lead to increased survival of individuals that contain these beneficial mutations. If the mutations are neutral, they don't have an effect on an organism's fitness. Silent mutations are usually neutral. And when the mutations are dangerous or deleterious, they decrease the fitness of the organism, and as a result, the organism has a lower chance of survival. Most mutations inside cells are neutral or slightly harmful. This concludes our section on how genes work. After going through this information, I'd like you to be able to respond to the questions listed on this slide. Thank you.